Hello everyone, my name is Tianduanyi Wang. I'm a PhD student from Aalto University and the University of Helsinki in Finland. Today I'm going to talk about the paper Modeling Drug Combination Effects via Latent Tensor Reconstruction. So first I'll give a brief introduction on drug responses. Drug response is the effect of a drug on a cell at a given concentration, and usually drugs are tested at multiple concentrations to get the dose response curves. The dose response curves are normally used to characterize the drug's cell killing effect, for example, by taking the half maximal growth inhibition point or the area under the curve. And high throughput drug response screenings are widely used in phenotypic drug discovery to find effective cell killing drugs. So we have single drug responses, or so-called monotherapy responses, and then we can also add two or more drugs on the cell and measure their cell responses. We will then get more complex dose response surfaces, which will require some specific synergy scores designed to evaluate the synergistic cell killing effect of two or more drugs in combination. A synergistic drug combination typically means that using two drugs together has greater benefits than using those two drugs separately. That's partially because two or more drugs usually target multiple genes and pathways. So drug combinations are promising in treating complex multifactorial diseases such as cancer and cardiovascular disorders. The combination therapy can enhance treatment efficacy and also overcome drug resistance. Thus, having wide clinical applications, especially in fighting cancer with poor prognosis and resistant to current therapeutics. However, due to the high dimensionality of the drug combination responses, where different drugs are tested at multiple concentrations in various cell lines, the experimental discovery of optimal combinations would be infeasible as the number of tested drugs increases. So machine learning methods are naturally a good choice since they are relatively time and cost effective to tackle this problem. So how did we compile our drug combination data? The data were taken from the NCI-Almanac project, and in order to match more cell features for the cancer cell lines, we took a subset of the NCI-Almanac data consisting of roughly 5,000 drug combinations against 19 different cancer cell lines. So in this data, as we can see from this three-dimensional cube, each data point in the cube indexed by drug 1, drug 2, and cell line represents a two-dimensional response matrix, as shown here, where the two dimensions are the concentrations of drug 1 and the concentrations of drug 2. So the three-dimensional cube together with the two-dimensional response matrix makes the data a degree 5 tensor. The 5 dimensions are the first drug, its concentrations, the second drug, its concentrations, and the cell line. It means that given a drug 1 and drug 2 tested in a cell line, and the concentration of the first drug and the concentration of the second drug, then we can get their combined response on the cell line. When the concentration of drug 1 is 0, then the combination response is actually a monotherapy response. So this is the structure of the drug combination data. And we encoded the structure of this fifth order tensor using one-hot encodings. And we also encoded drug structural information using max chemical fingerprints, where they are binary vectors indicating whether a substructure is present in a drug or not. And then we represented cell information using its multi-omics profile, including gene expression, copy number variation, CRISPR-Cas9 gene knockout experiments, and proteomic data. In order to reduce the large number of cell features, which is more than 70,000 in our case, we selected by only 1% of the omics features with the highest variance across the 19 cell lines. So the final feature matrix consists of the one-hot encoded tensor indices and the auxiliary information from both the drugs and cell lines. Due to the high dimensionality of the drug combination data, we can form different prediction tasks. For example, to learn missing entries or slices in the tensor. And different learning tasks also have different biological meanings. Here we investigated 3 different prediction scenarios. So given the index drug 1, drug 2, and a cell line, we can get a dose response matrix. For a drug pair, the first scenario is to predict the missing values in partially measured dose response matrices. And in the second scenario, we don't have the combined responses of this drug pair across all the cell lines, but only their monotherapy responses. So the task is to predict those combination responses with the known monotherapy information, along with the responses of each of the 2 drugs in combination with other drugs in the tensor. In the third scenario, we don't have the response information of this drug pair, including their monotherapy responses. And the task is to predict the complete dose response matrices based on the responses of each of the 2 drugs in combination with other drugs in the tensor. The first prediction scenario is considered easier since part of the combination responses of this drug pair already exist. And the second and third ones are considered more challenging, but also more practical, since normally we would like to discover novel synergistic drug combinations, and we don't have much information about this drug pair. So then how do we learn the combination responses? We built the ComboLTR method upon the latent tensor reconstruction-based polynomial regression method previously developed by our lab. So consider a polynomial of degree nd, which can be used to model nd-dimensional interactions. The polynomial functions have high representation power, but as the degree goes up, the exponential complexity will make it difficult to efficiently estimate all the coefficients. So here we represent the polynomial function in a tensor-based form, where T is the tensor for polynomial coefficients, and it can be decomposed into ND latent vectors. The general idea of this latent tensor reconstruction-based regression is basically that given a set of training data, we use a tensor representation for the polynomial functions, and then decompose and learn the latent parameters from training data, and then use the learned polynomial coefficients T to predict new drug responses. And then to simplify the computation with tensors, we can rewrite the tensor representation into inner products and then into pointwise products. Then we learn the latent coefficient matrices P and the projection matrix Q from the training data. The matrix Q is used to project the predictions onto the output space to tackle vector outputs. And the final optimization is to minimize the squared loss of measured and learned outputs and regularizations on the coefficient matrix P and projection matrix Q. And then we used a 5-fold cross-validation to evaluate the model performance of ComboLTR and compared it with 2 other methods, the random forest and ComboFM, which is a factorization machine-based method developed also by our lab. And the 2 figures on the left are the scatter plots of measured and predicted drug combination responses from ComboLTR and random forest as an example in the most challenging S3 prediction scenario. In this challenging scenario, ComboLTR clearly outperformed random forest. And the table on the right showed the predictive performance in terms of Pearson correlations when using different feature combinations. And it can also be seen that in the most challenging scenarios, ComboLTR benefited more from auxiliary features from different sources, such as drug and cell features. And due to the time limit of this presentation, not all results are presented here, but they can all be found in our paper. To further evaluate the predictive performance, We investigated the Pearson correlations separated for drug pairs in different drug classes and cell lines from different tissue types. The drug combination cell responses are grouped by their drug classes, whether the drugs are targeted therapies or chemotherapies, and by the cell's tissue types, whether the cells is from blood or breast. The two violin plots showed the Pearson correlations of predicted and measured combination responses in each tissue type and drug class for the most challenging S3 prediction scenario. And here the line means that some tissue types contain only one cell line. And in general, ComboLTR showed better and more stable prediction performance across different drug classes and tissue types in this challenging scenario, where less information was available. Next, we evaluated the predictive performance of the methods in terms of drug synergy. Here we use the NCI combo score to characterize the synergy of drug combinations. The NCI combo score is introduced by the NCI Almanac data publisher and is designed to evaluate the synergistic effects of drug combinations in this dataset. We calculated the NCI combo scores from complete, measured, and predicted dose response matrices, and used the scores to evaluate how well the methods can identify synergistic drug combinations. The two scatter plots above showed the correlations of measured and predicted NCI combo scores for ComboLTR and random forest in the S3 scenario. And we can see that ComboLTR clearly outperformed random forest with a much higher Pearson correlation. And we also did discrimination analysis using precision recall curves to further evaluate the model performance and classifying synergistic drug combinations. We labeled drug combinations as synergistic or non-synergistic based on different synergy thresholds on their NCI combo scores. The two figures below show the precision recall curves of ComboLTR and random forest with various synergy thresholds. ComboLTR had a much larger AUC, especially when recovering top synergistic drug combinations. And the above analysis together demonstrated that ComboLTR has much better performance in identifying highly synergistic drug combinations, even in the challenging scenarios. In addition, we also did some exploration on the cell omics features to investigate the contribution of omics data to the prediction. We did feature permutation to evaluate feature importance by randomizing each feature. We also extracted the coefficients of the polynomial learned by ComboLTR to inspect the feature weights. Besides this analysis, we tried also to include or exclude each type of omics data during training to evaluate its contribution to the prediction. In general, we found that the higher order tensor structure is dominating the prediction, especially in the easier prediction scenario. And due to the limited number of drugs and cell lines, here we have only 102 drugs and 19 different cell lines, the performance improvement brought by auxiliary features is also limited. But still, the method benefited from those auxiliary features, especially in harder prediction scenarios. So in summary, in order to search for optimal drug combinations from the vast combination space, efficient machine learning methods are needed. And we presented our ComboLTR method, which is based on representing high-degree polynomial regression models, and it learns a factorization of the parameter tensor containing the unknown regression coefficients. Thus, it can tackle large feature sets, including chemical and cell line features, from various data sources, and provide an efficient and practical way to predict novel drug combinations for preclinical and clinical evaluation. And more difficult prediction tasks, such as predicting novel drugs or cell lines outside of the combination tensor, are still to be explored. Auxiliary features might play a more important role in those prediction tasks. Besides, predicting combination responses with more than 2 drugs is also exciting and clinically meaningful. But due to the lack of enough training data, it still remains a future challenge. Okay, so this will conclude my presentation, and many thanks to all my colleagues and collaborators. And thank you all for your attention, and I'm happy to take any questions.